Before we look at today's lesson, let's just take a minute and review what you've looked at so far this week. So we are looking at eukaryotic gene expression and how our cells regulate that gene expression. On Monday's video, you looked at how cells can turn genes on and turn genes off at the chromatin level. We called that level 1 regulation. Yesterday's video looked at transcription initiation, so getting transcription started with all of those different types of transcription factors. We are calling that level 2 of regulation of gene expression. And then, like we talked earlier in the week, all the rest of this on this particular slide, we're going to group that into post-transcription regulation. We're going to look at a few different areas that fall in the category of post-transcription. We'll look at RNA processing. We will be looking at initiation of translation. So all this is going to be level 3 as well. And then we will look at protein processing. So we'll look at a few different types of regulation or areas that the cell can regulate gene expression. So these slides were from previous in the PowerPoint. I'm going to jump ahead to where we are today, which is going to be looking at post-transcription regulation. So all of this is, again, just to recap, is going to be all under level 3. The first one under level 3 that we're going to look at is RNA processing. So once the RNA is made, so once transcription happens and the polymerase uses the DNA code to create an RNA, you all know that in eukaryotes that RNA is processed. The first thing that happens, or one of the things that happen, is splicing. And you remember that that is removal of the introns and putting together the exons. This is an area that the cell can control, because you should also remember that at different times, different pieces of RNA can be spliced together to make different messages. So looking at this figure, here is our gene with 5 different exons. Remember, those are the ones that are expressed. And this whole sequence, the numbered sequences and the sequences in between, all get transcribed. But depending on what the cell needs, the exons can be put together in different combinations through splicing. So we can see that in this code, 1, 2, 3, and 5 were the exons that were used. They were spliced together to make this mRNA. This mRNA could then go out and be translated in the ribosomes, and we could make a protein product. So that would be one expression. This getting transcribed, and then it being processed into this message, that message going out and being used in the cell. But maybe the next time that this gene needs to be expressed, we want different exons together. So in this instance, we spliced together 1, 2, 4, and 5. So this mRNA would code for a slightly different protein product, which would have a slightly different function than the one that we did the first time. So the cell can control which option it uses. So this is an area of control, not necessarily turning a gene on or turning a gene off, but it's just an area that the cell can further control with their gene expression. The next part under post-transcriptional, so under our level 3, is mRNA degradation. And I just want you to think of this and kind of put this into perspective. Once an mRNA leaves the nucleus and goes out through the ribosome, it can be translated multiple times. So it's not like a one and done type thing where we make an mRNA and it's only used to make one protein. That's not how it works. An mRNA can go out of the nucleus and be translated over and over and over again. So some RNAs, depending on the 5' cap and the poly A tail, so you know those words, right? And this, they're calling it the leader and the tailor regions, but really that, or trailer, those are the 5' cap and the poly A tail. depending on how long specifically the poly A tail is, will tell the cell how many times to translate the mRNA. So if it's a really, really, really long poly A tail, that mRNA is going to be translated a lot, and the cell is going to end up with a lot of that particular protein. If the poly A tail is very short, the cell will only translate that a few times, and we will have less protein product produced from translation. So that is another area that the cell can control its gene expression. Again, it's not turning a gene on or turning a gene off. It's just able to regulate how much protein product is produced through translation. So our next one is initiation of translation. So again, we're still under level 3, but this is another place that the cell can control protein product production. This is looking at, does the cell translate mRNAs whenever they are available, or does the cell and do ribosomes translate RNA all at the same time? So for example, if we need a protein product like insulin, if we need that right now because you ate a really, really high carbohydrate lunch, your cells are going to express that gene, making RNA and then eventually making the protein insulin so that you can bring your blood sugar down. That is an immediate translation. Okay, so as soon as the cell makes the mRNA, it's going to be translated. But in other times, the cell wants different RNAs to all be translated at the same time and controlled. And for example, when the egg gets fertilized, so when we're making an embryo, initiation factors are the little proteins that need to be produced. And the mRNAs for those all are made, but then the cell kind of waits till they're all made and translates them all at the same time. So the initiation of translation can also be controlled by the cell. Can I just go ahead and translate the RNA whenever I need it? Or do I need to wait and translate it in a simultaneous, coordinated manner? That's what this one is. We're not really going to go into the mechanism of it. It does have to do with the 5' cap. That kind of serves as a message to the ribosome. Yes, translate me right now, or wait. But that's kind of just a general overview of this. The last area of control that the cell has is protein processing. It's kind of the first part, and then degradation. So protein processing. Once a protein is made on the ribosome, it's not necessarily completed, right? You probably remember the function of the Golgi body or the Golgi apparatus is to process and change proteins in certain ways by adding chemical groups to them or folding them properly. And that's the next area that we're going to look at. So cleavage, which means if we're going to cut it or if we're going to fold it, and then if the Golgi adds any chemical groups on there, is also subject to control. So again, it's not just something random. The cell has control over this process. So for example, if a gene for a membrane protein, so think like ATP synthase that's in the membrane, or one of those protein channels in the membrane, let's say we're trying to express that gene. That RNA is going to be translated on a ribosome, but then the ribosome will send its protein product to the Golgi. So the Golgi is going to make sure it's folded properly. If we're adding any like functional groups like methyl groups or acetyl groups or anything like that, carboxyl groups, if we're adding anything like that to the protein, the Golgi makes sure that those are added, and then it will sell those proteins to the membrane. It will take them to the membrane and make sure they're in the right spot. So that's what we mean by protein processing. We're making sure that the protein is folded properly and everything is added to it if needed. Also within this subject is protein degradation. So some proteins in the cell are very short-lived. The cell only needs to use them for a certain amount of time, And then when the function of that protein is done, the cell can kind of turn that protein off. Very similar to like the function of lactase in prokaryotes. Certain enzymes, certain things we don't need to have around all of the time, and our cells only make them when they're needed. So what the cell can do is they add something called ubiquitin, which is just a chemical marker to a protein, and that serves as a signal that says, hey, I'm no longer needed, you can digest me. So then these little, or I should say these big proteasomes they're very similar to like, I don't want to call them enzymes, but they're a protein structure that will degrade and break down that protein that we don't need anymore. So for example, if our body starts producing a digestive enzyme that we only need it for one meal, the cell can destroy that enzyme and get rid of it so that it's not floating around wasting space, wasting time, wasting energy in the cell. So that's our last part, is protein degradation. So take a couple minutes, maybe go back and review those. You'll notice that we didn't necessarily get into the technical details of what's all going on, but what I do want you to focus on is that the whole process of gene expression is controlled. It's not necessarily just turning genes on and turning genes off, but we can control the mRNA, we can control where it goes, we can control how long it survives in the cell, in the cytoplasm. We can control the processing and the shipping of proteins, and we can also control how long those proteins are present in the cytoplasm. So every step along the way, the eukaryote is able to control those steps. That's the big idea I want you to walk away with. Not necessarily the details of everything, but just that we have control over every single little step. In tomorrow's lesson, we'll look at one last topic, which is microRNAs. They are another area of control. But if you haven't done so far, make sure on that eukaryotic gene regulation worksheet that you have filled out levels 1, levels 2, and levels 3 so that you're not getting far behind, you're not getting confused. Have a good day. Talk to you soon.